So this is chromosomal theory of inheritance.
The right.
Yeah, this was given by Theodore Boveri and Walter Sutton. Chromosomal theory of inheritance. This was closely related to Mendel's work. Whatever Mendel had found, that same thing is related here. So it is just like parallelism to Mendel's finding. Whatever Mendel said is exactly shown by these two scientists. Just write down names. Okay. So what it is saying is, this chromosomal theory of inheritance, Sutton and Boveri in 1902 correlated Mendel's conclusion about genes. Mendel had found something about genes. Factors he used to say, or inherited traits to the behavior of chromosome during mitosis and meiosis. What happens to the chromosome during mitosis and meiosis were related to Mendel's work. Sutton is credited with first proposing the chromosome theory of inheritance. Now this you all know, chromosome always occurs in pairs. They are always in pairs. So total, if you consider human being, there are 46 chromosomes, 23 pairs. Homologous chromosomes separate during meiosis. Homologous chromosomes separate only during meiosis so that alleles are segregated. During meiosis or during gamete formation, the homologous chromosome, that is one parental, one maternal and paternal chromosome, they will separate. Meiotic products have one of the each homologous chromosome but not both. Okay. It will have one of the two chromosomes, not the both. What will happen after fertilization? The two chromosomes which were separated will again pair up. During meiosis, there will be only one of the homologous chromosome. But during fertilization, when the parent and mother and father's sperm and egg unite, you get the pair back. That pair is retained. Genes are located on the chromosome. Genes are located on the chromosome. This is what these scientists Sutton and Boveri found. Okay. So this can be asked for. Okay. Now chromosomal explanation of law of segregation. What is law of segregation? What is segregation? Separate. Okay. Means the genes separate during gamete formation. Okay. What is law of segregation? Two alleles of a gene that are found on a chromosome pair. Each gene has two alleles separate. Two alleles of a gene that are found on a chromosome pair separate, with the offspring receiving one from the mother and one from the father. That is law of segregation. Okay. Here you can see during G1 phase, it is chromosome. These are the two chromosomes. Okay. Now during G2 phase, you get duplicate chromosome. Because in between G1 and G2, which phase is there? S phase, synthesis phase. So the chromosome will get duplicated, extra more. Now during meiosis of anaphase, this will break. During meiosis of anaphase, this will break and you will get like this. And during meiosis 2 of anaphase, you will get separate. So in short, the chromosome is separate during, the genes separate during gamete formation. This is taken from NCERT. Then chromosomal explanation for law of independent assortment. You have to know what is law of independent assortment. Mendel's law of independent assortment states that the alleles of two different genes get sorted in gametes independently of one another. Now, alleles of two different genes, when we had studied dihybrid cross, it was round or wrinkled, yellow or green seeds. So finally, we found that we were getting round green or round yellow. We were getting round, which can be either green or round yellow. Or we were getting wrinkled green or wrinkled yellow. So the genes, the alleles are free to move anyway. Okay, it is not that if it is round green, it will be always round green. Round yellow, it will be always round yellow. You can get middle also, wrinkled green, wrinkled yellow, like that. So, of one. In other words, the allele a gamete receives from one gene does not influence the allele received from the other gene. They are free to move. The alleles are free to move only when there is dihybrid cross of two characters. Now this is what they are explaining. So here with colors we'll see. Now in first case, orange and short green. Orange and short green, the different alleles there. Orange is one. So you can get orange and short green, yellow and red. The other possibility is you can get orange and red and yellow and green. This is law of independent assortment. Possibility one is one long orange and short green, the way it is there, and one long yellow and one short red. Means normally it will be like this, or the second chance it can be, they can go to opposite friends. Like orange will go with green, orange will go with green genes, and yellow will go with red genes. That is law of independent assortment. Now, this is what we have to study today properly. Deviation from Mendelism. So Mendel had given 3 laws, but there were some deviations from Mendel's. Later further studies, they found that few characters are not following Mendel's law. Now, this is called as post-Mendel genetics or neo-Mendel genetics, after Mendel's work. So that's why it is called as post-Mendel genetics. Now, these are the differences or deviation: incomplete dominance or partial dominance, codominance, multiple factors of interaction of genes, epistasis or inhibiting genes, complementary genes, duplicate genes, multiple genes, polygenes, pleiotropism. What you have to know is definition, what is and 2 examples. Examples. Now let's go to the incomplete dominance of blending inheritance. 1:2:1 ratio it is said. Easy to understand. Incomplete dominance means the genes of the allele, allelomorphic is nothing but the genes of the allele are not related as dominance or recessive. They are not called as dominant genes or recessive. They express themselves when present together in the hybrid. So usually what happens if there is a dominant gene and if there is a recessive gene, which one will express? Dominant. But here what happens? Both will express. When in hybrid, if there is one recessive and one dominant, hybrid will obviously have one dominant and one recessive. Both of them will express, which was not seen in the case of Mendel. In case of Mendel, it was only dominant which will express. But here, both are expressing. Now see this. Example, F1 hybrid exhibit a character intermediate to the effect of 2 parent alleles. F1 hybrid. When we saw tall plant cross with dwarf, we got F1 generation, all were tall because it was capital T, small t. We saw in Mendel's case, when tall plant was crossed with dwarf, all the plants were tall. But here, the other characters, we are not seeing tall, some other characters of plants were not showing same pattern. It was different. Let's say example, inheritance of flower color in 4 o'clock. Very important, Mirabilis jalapa. Mirabilis jalapa and snapdragon dog flower. Now see this. What is happening here? What is the color of this flower? Red. What is the color of this flower? White. But in between, when you cross this red flower with white flower, the color changed to pink. When this pink color was self-crossed, what is self-cross? Crossing by itself. Taking 2 this type of pink color flowers and crossing. You got 1 red, 2 pink and 1 white. That's why it is called as 1:2:1. Okay. Red with white flower gave pink. There was no pink character. So this is blending. Both are expressing each other. See here, red and white. What you will get? This is P generation, mean parent. So we got F1 generation, all were pink. Intermediate phenotype is pink heterozygote. When pink was crossed with itself, you got red, 2 pink and 1 white. That is the example. Okay. So this is already seen that red and white gave pink color. The second example is Andalusian falls. What is fall? Good. Okay. 3 colors they are. Okay. Black, blue, and white. Black, blue, and white. Cross between pure black and pure white will produce hybrids. What will be the color of hybrid?
Pure white or pure black?
See, pure black and pure blue, and the other color is blue. Black, white, and blue. So the cross between pure black and pure white will produce hybrid. The color of the hybrid will be blue. There is no gray. What genotype and phenotype will you get if you self-cross the hybrid? Now you take the blue color. What genotype and phenotype will get? One black, one white, and 2 blue. Same thing. Okay. This is there. So here you can see black chicken when crossed with white, you got blue chickens. When blue chickens were self-crossed, you got 1 black, 2 blue, and 1 white. Now this is CBB, CWW. This is also a way to represent the genotype. But we have studied like this. We have taken capital B for black. Then what will be white?
WC.
Capital B is for black.
Capital W is black.
What we do? Small b. Then here what you will get?
Capital B is
Small b. When capital B self-crossed, you will get capital B, small b, and capital small b, small b. Okay. Next is Pisum sativum. That is peas. Okay. Pisum sativum, what you can see in this picture? Are they all of same size?
No.
Some are big, some are small, and some are intermediate. So this is what was found in Pisum sativum. Okay. BB stands for large size. A gene was there which was donated as capital B for large size. Then small b for use for small size. When they cross these two, they got intermediate. Okay, capital B, small b, which was it will be intermediate size.
So that P will come B.
That P will become middle of this. If one P is say 8 centimeter, the other P is say 3 centimeter, then it will be intermediate. Okay. The last part we'll see is for codominance. Now, what happens in codominance when the two alleles neither show dominant? Okay, they are not showing dominant, nor they are showing any recessive relationship, nor show intermediate condition. Not like your incomplete dominance. Incomplete dominance showed intermediate. Red and white. Obviously, we know red and white when we mix, we get pink color. That was shown in incomplete dominance. Here, what happens? It was neither showing dominant nor recessive, nor showing intermediate. But both of them were expressing separately. Both characters were expressed separately or simultaneously. Then the condition is called as codominance. Now, when you see this image, you can easily make out. These alleles are called as codominant. Now, white and brown. When you made them, you will get some white color and brown patches. So both are showing the characters. They are not blending, but both are expressing separately. So heterozygous show phenotype different from that shown by either of the homozygous. It will not show brown or it will not show white separately, but mix. Both the characters are there. Alleles are of same. Alleles, how you write? They are written with capital letter with different superscript. Now here you saw CW, CW, CR, CR. Color white, color white, color red, color red. That is what? You use one letter, but superscript you use for different color. That is one way to represent. Example RBC. ABO blood groups in humans. 3 blood groups in humans were discovered by Landsteiner. Which are the 3 blood groups?
A, B,
A, B, and O. Which is the fourth? The fourth group was discovered by Costello and Steeney, which is
B.
AB. Okay. This we'll see in the next class.
What is it?